lecture for Chapter 4 on the eukaryotic cell structure. In this class, we will be going over the different types of eukaryotic microbes. And the first group involve the fungi, which include the molds, which are multicellular, and the yeast, which are unicellular. But altogether, those are fungi. Also, a eukaryotic microbe group are the protozoa, and the protozoa are unicellular organisms that are found in the water and soil. Another group are the algae, and algae tend to be photosynthetic, and they can be unicellular or multicellular, and we often refer to the multicellular algae as seaweed. The last group of eukaryotic microbes that we're going to talk about are the helminths, and the helminths are worms. And worms technically are animals. They are multicellular organisms, multicellular eukaryotic organisms. And so in microbiology, we do talk about some animals, specifically the helminths. But the other group of animals that we're going to be talking about a little bit at the end of the semester are humans. Specifically, we will go over the immune system. And the reason the human immune system is part of microbiology is because the human immune system evolved to protect us from all these different types of microbes. As I go through this lecture, I want you to pay attention to two different things. The first thing I want you to pay attention to is understanding the differences between the eukaryotes and the prokaryotes. And this is really important, especially in healthcare, because when a person gets sick, that person is composed of eukaryotic cells. So we technically are animals, and all of our cells are eukaryotic cells. And if we get sick with a bacterial infection, the bacteria is a prokaryotic cell. So when you have an individual who is sick with a bacterial infection, to cure that person, what you want to do is you want to be able to kill the prokaryotic cells, but not have that drug harm the human cells. So we call that selectively toxic. So the best type of drug will be a drug that will kill the prokaryotic cells and leave the human cells alone. So it's not toxic at all to the humans. And that's very important. And the way you can find those types of medication is by understanding the differences between eukaryotes and prokaryotes. So you can find structures, you can find processes that are specific to the prokaryotes and have drugs that target those structures that are not found in eukaryotic cells. And again, we call that selectively toxic. So that medication is toxic only to the prokaryotes and it does not harm the eukaryotes. The other thing that I want you to pay attention to during this lecture is identification. So how are we able to identify different eukaryotic cells from each other? Because people, when they get sick, they're not only infected with prokaryotes, they could be infected with eukaryotic microbes. One example could be this amoeba. This is Entamoeba histolytica, and it is on your microbes to memorize list, and it causes amoebic dysentery. Another eukaryotic microbe is Giardia lamblia, which causes giardasis. And so these are two different types of eukaryotic microbes. And if you have an individual who is sick, you need to know which type of microbe is infecting them in order to be able to treat them effectively. In general, eukaryotes, especially when compared to prokaryotes, they are much larger cells. So they're very large and they are complex. And in terms of complexity, they have organelles. And so that's very different than the prokaryotes. Remember, the prokaryotes are very small and they're very simple cells and do not have organelles. Also, eukaryotic organisms can be unicellular or multicellular. So unicellular means that an organism is composed of one cell. Multicellular, the organism is composed of many cells. Also, eukaryotic microbes can be heterotrophic or autotrophic. Heterotrophic means other feeder, means that in order to get organic molecules, they have to eat another organism. Autotrophic, auto is self-feeder. That means they can make their own organic molecules, so basically they are photosynthetic. So eukaryotic organisms can be either heterotrophic or autotrophic. Also, in terms of reproduction, eukaryotes can reproduce asexually or sexually. So with the eukaryotes, you get a lot of variety in their characteristics. This is a diagram of a typical eukaryotic cell. Well, typical half of it is a typical plant cell and half of it is a typical animal cell. And you should know that plant cells have some structures that animal cells do not. Plant cells have a cell wall, a rigid cell wall. They also have a central vacuole and they will have chloroplasts. So those are structures that the animal cells do not have. And actually in this class we will not be talking about plant cells because technically they are not part of the eukaryotic microbe group. But we will be talking about animal cells. So what I want to do next is go through the basic structure of a eukaryotic cell. And I'm going to start on the outside of the cell talking about the appendages first. Then I'll go through the layers of the cell and then I'll talk about what you find inside the cell. The first appendage that I want to go over are the flagella. Flagella is plural, flagellum is singular. It's good for you to try to use the correct plural and singular form of these scientific terms, but don't worry, I don't take points off if you use the wrong one. So a flagellum is relatively long compared to the length of the cell, and there are relatively few on the cell. And their purpose is locomotion, so getting the cell from one place to another. And that is the same purpose that you have in the prokaryotic cells for their flagella. There is a difference in structure between eukaryotic and prokaryotic flagella. Usually the eukaryotic flagella are much more complex in their structure compared to the prokaryotic flagella. So their function is the same, but the structure is a bit different. So if you were to just look at the structure of a flagellum, you would be able to identify if it came from a eukaryotic organism or a prokaryotic organism. The second type of appendage you can find on eukaryotic microbes are cilia. And cilia are much shorter than flagella and they are very numerous on the cell. And the purpose of cilia is movement. And it can be locomotion, it can be used to get the cell from one place to another, but it can also be used to move fluids. So one example is in the human respiratory system. You have ciliated epithelial tissue, so here are the cells, and here's the cilia on the ends of the cells. And obviously these cells are not going to swim around. So this cilia is not used for locomotion for these cells. Instead, it is used to move mucus. So in the human trachea, you have the respiratory epithelium, which is a ciliated epithelial tissue. So here are the cilia, these blue fibers here that are attached to the cells in the epithelial tissue. And on top of the cilia, you have a layer of mucus. So this gray layer of mucus is laying on top of the cilia. And the purpose of the mucus is to trap any particles, microbes that a person inhales. So as a person inhales air, if there are any particles, those particles are going to get trapped in the mucus, and then the cilia beat to move the mucus away from the lungs. And that helps to prevent infection. So in this case, the cilia are involved in movement, but it's not locomotion. These cells are not moving anywhere. Instead, the movement is to move a fluid. So there are only two types of appendages you may find on a eukaryotic cell, flagella and or cilia. And remember, prokaryotic cells, they can have flagella, fimbriae, and or pili. Now what I want to do is go through the different layers of the cell, of the eukaryotic cell. The outermost layer that is found on most eukaryotic cells is the cell wall. And the purpose of the cell wall is structure, to hold the shape of the cell. And we are probably most used to thinking of cell walls on plant cells. Now, compared to the prokaryotic cell wall, the eukaryotic cell wall actually has a very simple structure. So it's very simple compared to the prokaryotes. Prokaryotic cell walls are much more complex. But there are different components in eukaryotic cell walls that will help you identify different eukaryotic organisms from each other. Fungi have cell walls, and when you look at the structure of a fungal cell wall, the main component you find is chitin. And chitin is the type of carbohydrate that is indigestible. Our digestive systems are unable to break it down. And chitin is also found in the exoskeletons of insects. That's a cicada exoskeleton. And also in the shells of crustaceans, so lobsters and shrimp. So again, that is chitin, and that is very characteristic of a fungal cell. Now remember, fungi, they can come in two forms, mold form and yeast form, and there is a difference between mold and yeast cell walls. Yeast cell walls have mannan and glucan. So this would indicate that this must be a yeast cell wall because it has chitin, mannan, and glucan. If it was a mold cell wall, it would only have chitin. Algae also have cell walls, and the main molecule that makes up the algal cell walls is cellulose. So cellulose is found in a couple different forms in algae. And cellulose is the same molecule that you find in plant cell walls. So again, it's another type of carbohydrate, and you know that because it ends in -ose. So that's one way you can identify algae from fungal cell walls. Fungi have chitin, algae have cellulose. Protozoa are a little bit different compared to the other two eukaryotic microbes in that they do not have a true cell wall, instead they have a pellicle. And a pellicle is composed of protein, which is another difference. The others are composed of carbohydrates. And the pellicle is a thick protective outer covering. So this is the pellicle that surrounds protozoa. All right, so the components that make up the cell wall can be used for identification. Fungi have chitin in their cell wall, algae have cellulose, and the protozoa have a pellicle composed of protein. And remember, this is all compared to prokaryotes, bacteria that contain peptidoglycan in their cell wall. Animals are the only type of eukaryotic organism that do not have a cell wall. And instead of having a cell wall, they have a glycocalyx. And this was a term that I also talked about in the prokaryotes. And remember, glyco refers to sugar, calyx refers to coat. So it is a sugar coat that is the outer surface or the outer layer of an animal cell. So this is the glycocalyx. And in the glycocalyx, you have lots of glycoproteins, glycolipids, again, some sort of molecule attached to a carbohydrate. And the main purpose of the glycocalyx is similar to the purpose of the glycocalyx in the prokaryotes in that it is important for attachment. So it's important for the cells to attach to surfaces and attach to each other. And also the glycocalyx for a eukaryotic cell, it helps to strengthen the cell. And most importantly, the glycocalyx is involved in cell-to-cell recognition. So this is how different eukaryotic cells are able to identify each other. And this will become very important when we talk about the immune system, specifically when your white blood cells are identifying cancer cells or they are identifying cells that are infected with a microbe. The last layer of the eukaryotic cell that I'm going to talk about is the plasma membrane, and the plasma membrane serves the same function as it does in a prokaryotic cell or in any cell. So the purpose of the plasma membrane is to be a semi-permeable barrier. So it separates the environment from the inside of the cell. And the basic structure of the plasma membrane is very similar to the prokaryotic plasma membrane in that the main matrix is a phospholipid bilayer. So if you remember, a phospholipid is composed of a hydrophilic head or a polar head and two hydrophobic or nonpolar tails. And when you mix those phospholipids together with water, they will automatically form a bilayer, two layers, one layer of phospholipids here, the other layer of phospholipids here, and their hydrophobic tails are forced together. So again, the phospholipid bilayer makes up the main matrix of the plasma membrane. And a eukaryotic plasma membrane will also have proteins, different types of proteins embedded in the membrane, but these proteins are usually not enzymes. Prokaryotic cells more often have enzymes embedded in the membrane. Eukaryotic organisms have very few enzymes embedded in the membrane. Most of the eukaryotic enzymes are in the organelles. And another difference between a prokaryotic and a eukaryotic membrane is that eukaryotes have cholesterol embedded in the phospholipid bilayer. Prokaryotic cell membranes do not have cholesterol. And humans, animal cells have cholesterol, and there's a difference between animals and fungi. Fungal cells have a different type of sterol that's called ergosterol. So these sterols, they help to stabilize the structure of the membrane. And so cholesterol is found in animal cell membranes and ergosterol is found in fungal cell membranes. So I've gone through the appendages of a eukaryotic cell, so flagella and cilia. I've also gone through the layers of a eukaryotic cell. So cell wall is found on most eukaryotes, in fact, almost all of them, except for the animal cells, and instead the animal cells will have a glycocalyx as the outer layer. But then all eukaryotic cells will have a cell membrane. So all cells must have a cell membrane, and those are the layers of the eukaryotic cell. Now what I want to do is go through what you will find inside the cell. So once you cross over into the membrane, what you find inside the cell. First, I want to go through a little bit of vocabulary. There are two words, cytoplasm and cytosol. And often in a lot of biology classes, they are used interchangeably, but they're not exactly interchangeable. When you say the term cytoplasm, you mean everything in the cell between the cell membrane and the nucleus. So everything between this is considered the cytoplasm. So that includes the liquid inside the cell and the organelles. So technically the cytoplasm is everything inside the cell except for the nucleus. Cytosol, this specifically refers to the liquid inside the cell. So if you're just referring to the liquid, you want to use the term cytosol. So to go through the cytosol in a eukaryotic cell, it's basically the same as a prokaryotic cell. It's mainly water and it has all sorts of dissolved nutrients, dissolved waste products, dissolved sugars, salts, ions, that sort of thing. And the only difference is that a eukaryotic cytosol is about 90% water. Compared to the prokaryotic cell, the eukaryotic cell is going to be hypotonic. And again, that's important when you're looking at osmosis, when you have a prokaryotic cell living inside a eukaryotic cell. In addition to the cytosol, you will also find a cytoskeleton inside a eukaryotic cell and throughout the whole eukaryotic cell. And the cytoskeleton is made of all sorts of different protein fibers. Larger ones are called microtubules and the smaller ones are microfilaments. And so the cytoskeleton is a lot more complex in a eukaryotic cell than it is in a prokaryotic cell. And the cytoskeleton has several different functions in a eukaryotic cell. And one of the first functions is that it holds all the organelles in place. Sometimes I think we think that the eukaryotic cell is like a swimming pool with a lot of balls thrown in it for the organelles, and everything is free floating and moving around just randomly. That's not the case. The cytoskeleton actually holds all the organelles in a particular place. So everything in there is not just free floating, it's actually held in place, and the cytoskeleton does that. Another function of the cytoskeleton is to form the centrioles and the spindle fibers. And centrioles, animal cells have two centrioles, and the centrioles, if you look at a cross section, they have a very complicated structure. And like I mentioned before, the prokaryotic cytoskeleton is not very complicated. But the main purpose of the centrioles and spindle fibers occurs during mitosis. So mitosis is the type of asexual reproduction that eukaryotic cells go through. And you should know the different stages of mitosis. Begins with prophase, then metaphase, anaphase, and telophase. And you should know what is happening during those different stages. When the cell is getting ready to divide, and then finally telophase, when you have the two sets of genetic material that have separated, and then the cell begins to separate itself or divide itself, and that's cytokinesis. Well, the centrioles, they organize the spindle fibers. And here's a better picture of spindle fibers during anaphase. The blue structures are the chromosomes, and then the red are all the spindle fibers, which are composed of cytoskeletal fibers. And what those spindle fibers are doing is they're pulling the two sets of chromosomes away from each other so that they can unwind at two separate poles, and then you can get cytokinesis between those two sets of genetic material, giving rise to two identical daughter cells. Another purpose of the cytoskeleton is to perform or facilitate cytoplasmic streaming. And cytoplasmic streaming is when the cytosol is circulating in the cell. So cytosol, remember, that is the liquid part of the cell. And when these different types of cytoskeletal fibers, microtubules, microfilaments, when they contract, that gets that liquid circulating all around the cell. And the purpose of cytoplasmic streaming, there are a couple of different purposes. One is to distribute nutrients. Eukaryotic cells are relatively large, and so if the eukaryotic cell had to wait for nutrients to go from one end and just diffuse to the other end, it would take too long. So to facilitate movement or transport of nutrients, you have this cytoplasmic streaming that will get the nutrients distributed around the cell much faster than simple diffusion. Another purpose of cytoplasmic streaming that you see in protozoa and animal cells is to form pseudopods. So pseudopods, pseudo-fake, pod feet. This is how some cells move. So what happens is that the cytoplasmic streaming, it will move the cytosol towards one end of the cell, pushing out that end of the cell, forming a pseudopod. And then it can change direction as the cytoskeletal fibers contract and relax, and it can push out in another part of the cell, creating another pseudopod. And basically that's how amoebas move. So that crawling around on pseudopods, that is facilitated by cytoplasmic streaming, which is done by the contraction of the cytoskeleton, moving that cytosol. The last function of the cytoskeleton is transport of material into and out of the cell. Transporting material into the cell can be done by endocytosis. Endo means inside, so the cell brings material inside the cell. And this involves moving the membrane. So the membrane actually has to move, and the only way you can move the membrane is by using the cytoskeleton contracting the fibers to move the membrane. And there are two types of endocytosis that you may have heard of before. phagocytosis and pinocytosis. So phagocytosis, phago means to eat. So phago means to eat, so that is when the cell brings in a solid particle. The other term is pinocytosis, and pino means to drink. And pinocytosis is when the cell brings in a liquid nutrient. But they're both types of endocytosis. But an important type of endocytosis that you need to know for this class is receptor-mediated endocytosis. And receptor-mediated endocytosis means that the process of endocytosis is triggered when a particle binds to a receptor. So when a particle binds to a particular receptor, that triggers this process of endocytosis, forming a vesicle with the particle bound to the receptor, and it's inside the cell. And this will become very important next week when we talk about viruses, because this is how some viruses trick the cell into getting into the cell. The virus binds to the receptor, the cell thinks it's a food particle, and it performs endocytosis, and that's how the virus gets inside the cell. And in addition to endocytosis, exocytosis is the opposite. Exo means outside. That's when the cell releases particles outside of itself. And again, it involves moving the membrane, so it involves the cytoskeleton. Now the organelles. And before I go through the different types of organelles, I just sort of want to define the term organelle. An organelle is a membrane-bound area of cytoplasm, so it has a membrane. And all organelles have a specific shape and function. So organelles are membrane-bound areas of cytoplasm that have a specific shape and function. So organelles they all must have a membrane. They all have a particular shape. So the nucleus has a particular shape. You can identify the nucleus from like a mitochondria. And the organelles all have a particular function. And the reason the organelles have their own separate function is because they contain a specific set of enzymes. And remember, I told you that eukaryotic cells tend to keep their enzymes in the organelles, where prokaryotic cells do not have organelles, so their enzymes are kept in the membrane. The first organelle that we always go through when we talk about eukaryotic cells is the nucleus. And the reason for this is because the nucleus is usually the largest organelle and the most visible. Remember when I talked about naming prokaryotic cells and eukaryotic cells were named true kernel cells because when they started looking at living cells, they saw these large cells that had a kernel in them. So that's, of course, where the name eukaryote comes from. The purpose of the nucleus is to protect the DNA. So that is what you find inside the nucleus is the DNA. And when the DNA is complexed with proteins called histones, it's called chromatin. So the purpose is to protect the DNA. The nucleus itself has a double membrane, so two-layer membrane. And also in the membrane, you have these pores, these holes, so these nuclear pores. And that allows transport into and out of the nucleus. Now, the important thing about the transport is that this DNA that is inside the nucleus, it never leaves the nucleus. Because remember, the purpose of the nucleus is to protect the DNA. So if the DNA could be transported out, then that would negate the purpose of the nucleus. So DNA never leaves the nucleus. But what does leave the nucleus is RNA. And RNA can leave the nucleus. Remember, during transcription, you get mRNA produced, which is a copy of the DNA, and then that has to leave to interact with the ribosomes and be translated. Also inside the nucleus, you have the nucleolus. So the nucleolus is where rRNA is made. And the little r stands for ribosome, and the rRNA is part of the structure of the ribosome. And so this also has to be transported out because the ribosomes will be found on the rough ER and in the cytoplasm. So RNA can be transported out, and actually some proteins can be transported in. So various types of proteins, various types of enzymes can be transported in. So enzymes, proteins can go in, RNA can come out, but DNA stays inside the nucleus. And the nucleolus is technically not an organelle. It is a darker staining part of the nucleus. And it's not an organelle because it doesn't have its own membrane. Remember, the definition of an organelle is a membrane-bound area of cytoplasm that has a specific shape and function. And of course, prokaryotes do not have a nucleus, but they have a nucleoid, and that is the area where you find their chromosome. Speaking of chromosomes, remember I told you that prokaryotes have a single circular double-stranded chromosome, which is composed of DNA only, and that is different than eukaryotic chromosomes. Eukaryotes have multiple chromosomes that are linear, so they are linear like lines, and they are a complex of DNA, and histones. So histones are a type of protein. So the DNA is wrapped around these complexes of histones. And that is what we call chromatin. So this complex of DNA and histones is chromatin. And chromatin means colored body because that's what they could see. When they stained it, they could see these colored bodies, and we found out later their DNA wrapped around protein. The next organelle that we always talk about is the ER, the endoplasmic reticulum. And the endoplasmic reticulum is actually an extension of the nuclear envelope. So here in this diagram, it's really nice. It shows the nuclear envelope, the gray nuclear envelope, extending out into the green ER. So the ER is always associated with the nucleus because it's an extension of the nuclear envelope. And there are two types of ER, the smooth ER and the rough ER. And the purpose of the smooth ER, and this is the smooth ER, so it's smooth, it doesn't look bumpy. The smooth ER has all sorts of proteins embedded on it that will make the fats and the steroids for the cell, and also the phospholipids. And in some cells, it has enzymes that can detoxify the cell. So that's the purpose of the smooth ER. The rough ER is called rough ER because it has ribosomes embedded in it. And so these little red bumps, these are the ribosomes. Ribosomes are very important, and all cells must have ribosomes. And ribosomes are not considered organelles. So they are enzymatic complexes, like big complicated enzymes. And they are composed of protein and rRNA. And remember, the rRNA is made in the nucleolus. And the ribosomes can be found bound to the rough ER or floating free in the cytoplasm. And the purpose of the ribosome is translation. So they take the mRNA and create the protein product. And eukaryotes and prokaryotes both have to have ribosomes. The difference is that the prokaryotic ribosome measures 70S, but the eukaryotic ribosomes measure 80S. And the importance of the measurement, it has to do with density and size, but the point of this is that you can identify prokaryotic ribosomes from eukaryotic ribosomes. So again, prokaryotic ribosomes measure 70S, eukaryotic ribosomes measure 80S, but they perform the same function in the cells. The next organelle is the Golgi complex or the Golgi apparatus. I just tend to call it the Golgi. And the Golgi is closely associated with the ER, and it looks like a series of sacs, sort of flattened sacs. And the purpose of the Golgi is to take the products of the ER, to modify those products, especially proteins that are produced by the rough ER, to modify them into a functional form, and then to package those products into vesicles, little membrane-bound areas, so sort of like little membrane bubbles, to package those proteins or those products into vesicles, and then often those vesicles are transported to another part of the cell, or they are transported out of the cell by exocytosis. One product of the Golgi is another organelle called a lysosome. And a lysosome is a vesicle produced by the Golgi that contains digestive enzymes. And the purpose of the lysosome is to act as like the stomach for the cell. So if the cell performs phagocytosis, it takes in a solid food particle, forms a food vacuole with that solid particle in it, then that vacuole will fuse to a lysosome. Once it does that, all these digestive enzymes will break down that food particle. So break it into the little pieces so it can be absorbed and used as nutrients for the cell. So phagocytic cells often contain a lot of lysosomes. And another thing about the lysosome is sometimes it has been referred to as a suicide sac. Suicide sac. And the reason for this is because this lysosome ruptures, and these digestive enzymes are released out into the cytoplasm, then those enzymes can start digesting the other organelles for the cell, the mitochondria, the ER, the nucleus, and that will result in the death of the cell. So that is a type of death called apoptosis. So basically cell suicide. And when cells die by apoptosis, then that in a multicellular organism like a human, then you do not get inflammation. And that happens a lot during development. So when you were a fetus, at one point you had tissue between your fingers. We had webbed fingers. Well, at some point those cells got the signal to go through apoptosis. Their lysosomes ruptured, those cells died back, and so now when you were born, you did not have that tissue between your fingers. So that happens a lot during fetal development. Peroxisomes are another type of organelle that are similar to lysosomes. They are a vesicle that contain different types of enzymes. And some of these enzymes are used to oxidize amino acids and fatty acids. But they also contain an important enzyme called catalase. So catalase, of course, is an enzyme, and this enzyme breaks down hydrogen peroxide, H2O2. And hydrogen peroxide is actually toxic to cells because it will oxidize DNA and oxidize enzymes. So the cell needs to protect itself from the peroxide, hydrogen peroxide, so the catalase will do that. Mitochondria are known as the powerhouse of the cell, and they perform aerobic respiration. And the purpose of aerobic respiration is to take glucose and to run it through a series of reactions to produce ATP. And you should know that ATP is the usable form of energy that a cell needs to function. So all living cells need to produce ATP. So that's the purpose of the mitochondria. In terms of the structure of the mitochondria, all mitochondria have this sort of bean shape, this sort of curved oval bean shape, and all mitochondria have two membranes. They have an outer membrane and an inner membrane. And the outer membrane has a structure very similar to the plasma membrane, the eukaryotic plasma membrane. The inner membrane is a little bit different. The inner membrane has all these cristae, and the cristae are all these folds, so it has a lot of surface area. And the other thing about the inner membrane is that it has a lot of enzymes embedded in it. So a lot of enzymes. Another thing about the mitochondria is that all mitochondria have their own DNA. So they are actually an organelle that contains DNA, and the form the DNA is in is they have a single piece of circular DNA that is double-stranded. The last unusual thing that the mitochondria have is that they contain their own ribosomes. So they have their own ribosomes, and if you look at the ribosomes, they measure 70S. So these are some characteristics that are kind of unusual about mitochondria. So mitochondria, the inner membrane, has a lot of enzymes, which is very similar to a prokaryotic membrane. They also contain their own DNA, and that DNA looks like a prokaryotic chromosome. And they also contain their own ribosomes, which measure 70S, which look like prokaryotic ribosomes. And the other thing about the mitochondria is that they divide like prokaryotes divide. And that process is called binary fission, which is very different than mitosis. So mitochondria look like primitive bacteria. The last organelle I want to talk about are the chloroplasts. And chloroplasts are only found in photosynthetic organisms, so you do not find these in animals. But the purpose of the chloroplast is to perform photosynthesis. And you should know that photosynthesis is where the organism takes light energy and carbon dioxide and produces glucose. So that's the purpose of photosynthesis. And if you look at the chloroplast, you see that it has a lot of similarities to the mitochondria. First thing is that it has two membranes. It has an inner membrane and an outer membrane, which is similar to the mitochondria. Also, when you look at the chloroplast, it has its own DNA, and it is a small circular piece of DNA that is double-stranded and DNA only. Also in the mitochondria, it's here already, they have their own ribosomes, and those ribosomes, guess what they measure? They measure 70S. And the other thing about the chloroplast is that they divide. They can divide like bacteria, again, a binary fission. So the chloroplasts also look like primitive bacteria. The fact that mitochondria and chloroplasts look like primitive prokaryotic cells led scientists to develop an idea, a hypothesis called endosymbiosis. And you should know that symbiosis is when two organisms live in close contact and they evolve together. Endo refers to one organism living inside the other. And this idea of endosymbiosis, sometimes called the theory of endosymbiosis, so a theory means it's well accepted, pretty much accepted as fact. The idea of endosymbiosis is to describe how eukaryotes evolved from prokaryotes. So during evolution, way at the beginning of evolution, they think that the Earth is about 4.5 billion years old. That's when the Earth was formed, 4.5 billion years ago. And about 3.5 billion years ago, the first cells evolved. And these cells would have been probably the simple prokaryotic cells. And you would have had a lot of variety with the prokaryotic cells. There would have been big ones, there would have been small ones. And what do big things usually do to small things in nature? They usually eat them. So what would have happened is at some point, this large cell would have phagocytosed the smaller one. And so eventually, you would have had a large cell engulfing a smaller one. And that would have led to the formation of mitochondria and chloroplasts. This is a nice diagram that goes through a little bit more of endosymbiotic theory if you're interested. And basically, endosymbiotic theory starts with a prokaryotic type of a cell. So it has a cell wall, it has DNA, and that's pretty much about all it has. At some point, the cell wall is lost, so it's making it look more like an animal cell. And then the membrane folds in, it invaginates, and eventually enough to form the nuclear membrane, and then lead to the ER and probably the Golgi apparatus. So that initially what was happening, that infolding invagination of the cell membrane led to the nuclear envelope, the ER, and the Golgi apparatus. And after that was done at some point, this cell that is evolving into a eukaryotic cell would have phagocytosed a bacterium that could perform cellular respiration, aerobic respiration, and that would have developed into the mitochondria. And later on during evolution, one of these cells would have phagocytosed a photosynthetic bacterium, and that would have led to the development of the chloroplast. So the formation of the first eukaryotic cell would have happened about 2.5 billion years ago. And for this to happen, you would have had that infolding of the membrane forming the nuclear envelope, the ER, and then at one point, that larger cell would have phagocytosed a little bacterium that can perform cellular respiration, and eventually they worked out a symbiotic relationship where this cell would have provided ATP for the parent cell. And the evidence, of course, for this is that you have two membranes. The mitochondria also has its own ribosomes that measure 70S. The inner membrane has a lot of enzymes. It also has its own circular piece of DNA, and it can divide like a prokaryotic cell. Later on, a descendant of that cell that worked out a symbiotic relationship to form the mitochondria would have phagocytosed a photosynthetic prokaryote, and over time that would have developed into the chloroplast. And again, the evidence for this is the double membrane, where the outer membrane is very similar to the cell membrane of the eukaryotic cell. The inner membrane has lots of enzymes, which is similar to a prokaryotic cell. The chloroplast has its own 70S ribosomes, has its own circular piece of DNA, and it divides very similar in method to a prokaryotic cell. All right, so that was the overview of eukaryotic cell structure. So make sure you know all the different parts of the eukaryotic cell and that you can compare and contrast the eukaryotic cell to prokaryotic cells.